Hi, this is John Horgan, science correspondent for Blogging Heads TV. And with me today, we have a very special guest. And I wonder if you could introduce yourself to the Blogging Heads audience. Special guest.
Thanks very much, John. I am a Neuroskeptic. I'm a pseudonymous blogger. I blog about neuroscience, psychology, and other issues for Discover magazine.
Hmm.
And I've been writing my blog under a pseudonym for 7 years.
Wow, that long. Okay, I didn't realize it had been that
Oh
long.
yeah, it's a long time now.
Yeah. So could you just briefly tell us why you became a neuroscientist in the first place, and if you have a specialty, tell us a little bit about that.
Hmm. Um, well, I'd always wanted to be a scientist. I'd always wanted to do research,
Hmm.
even from when I was very little. I think when I was at school and people asked me, "What do you want to be when you grow up?" My answer was that I wanted to be a mad scientist. Um, and so I'd always wanted to be a researcher, but I didn't really have a particular interest in neuroscience actually until my second year of university, when I took a course in it and a course in psychology as well. And I was very grabbed by it, and it struck me as being much more um, exciting than other areas like biochemistry, chemistry, which I'd always been focusing up to them, but they didn't really seem to have very much of the kind of unknown factor
Hmm.
anymore because they were sort of too well understood already. Whereas in neuroscience and psychology, it's much more of um, you know, there are huge things remaining to be discovered.
Was there any particular, I mean, neuroscience, psychology, that is a vast realm. Was there any particular
Mm.
problem or area of expertise that you thought you would focus on?
Um, Actually, funnily enough, not really. I was never really, and still I'm not really kind of focused on one particular area of neuroscience. I mean, my current research is using neuroimaging methods to measure and investigate transmitter systems in the brain,
Hmm.
neurotransmitters. Um, but that's not um, to say that I'm not interested in or fascinated by other areas of neuroscience. It's really the whole field is of interest to me.
And at what point, what inspired you to start writing a blog called Neuroskeptic? I mean, why that
Yeah.
particular title?
Um, so this was a long time ago, but I still remember the day I decided to start my blog. I'd been so I was in the middle of my PhD at the time, and I'd been concerned for a while about what I was doing, in the sense that I had growing concerns that the way in which I'd been taught to approach um, neuroimaging and fMRI, in particular, um, was not really scientifically or statistically um, rigorous. And but this was not just me and not just people around me; it was the whole field.
Hmm.
So I was sort of growing disillusioned, if you like, in what I was doing myself, but also in the field. And I was worried that what I was doing at the time was, not really rigorous, scientifically justified um, investigation to answer a particular question. It was fishing for positive results and continuing to fish until you find them, and then coming up with a narrative after the fact, post hoc explanation of what you happen to find. And I was aware that I was doing that myself, and also that a lot of other people were doing it. So I decided that the best way to approach that was with a blog. The particular name I chose, I think it was inspired by the Neurocritic blog,
Right.
which had been around for a couple of years at that point, and it's still going. So that was an inspiration for me and for the name. But I think I was also inspired by, there's a book called The Skeptical Chemist,
Mm-hmm.
or spelt CHY Mist. And I forget who it was. This was in like the 17th century. And this was one of the earliest books in chemistry, because before that, everything was just like alchemy and complete rubbish, basically.
Uh-huh.
And this book came along, and it was The Skeptical Chemist, and it was saying, a lot of this stuff is not very good. But that in itself, even though it was skeptical, but it laid the foundation for better stuff. So it's not just destructive, but also constructive at the same time.
Right. Why anonymous?
Mm. That's probably the single question that people ask me most often, and I never really have a very good answer for it. At the time when I started it, I was worried that what I was going to be saying was very controversial and very unpalatable to other people. So I thought that if I said this under my own name, it might be annoying a lot of people and perhaps making it difficult for me to finish my PhD. And I think actually, in retrospect, that was actually completely an unfounded fear, because actually many people have had the same concerns. And I wasn't the first person to have them, or even the first person to say them.
Mm-hmm.
But it seemed like that at the time. It seemed like no one was talking about these things, but I think actually they were. But I wanted to make it more, say it louder, if you like, than other people at the time. So that was the feeling when I started, and then since then it's really become part of the identity of my blog, is that it's pseudonymous.
Right.
But I think that nowadays, most of what I say, most of what I write about, is probably not that controversial, in the sense that, although it may be controversial that I'm saying it in the way that I say it, but the actual the substance is pretty well understood at this stage. Yeah.
Well, I have to say that I think you have a knack for, and I say this as somebody who is a critical science reporter. You have a knack for saying what is actually quite pointed criticism in a way that doesn't sound nasty, and so
Mm,
it might be more. I think the kinds of the way that you put things actually might change more minds than a more sort of
mm,
aggressive, uh, snarky style than my style, for example. And so that I really admire the way you talk about some of these issues, and I'm
mm,
concerned with some of the same things that you are, and I've
mm,
written about them. Yeah, I would think that if you decided to tell everybody who you were, that could only benefit you as a scientist, because you are obviously a constructive critic, not somebody just trying to tear the
mm.
field down for its own sake. So,
Yeah. I mean, so it's interesting that you said that my style is kind of more constructive. I think that's something I've always tried to do, and I've always been guided by the feeling, as I said, that, and this was right from when I started my blog, the feeling that I'm not saying that I'm immune to any of these things. I'm not trying to say that I'm better than anyone else, any other researcher, and I'm not trying to say that I'm doing things the right way and everyone else is doing things the wrong way. Because in fact, I'm well aware that in my own research, I'm not saying that I'm doing things, as if in the ways that I say that things should be done always.
Right.
So in that sense, I have no real. I'm not on a high horse, and I know that I'm not, and so I don't feel that I'm able to say, be nasty or even just be what's the word, imperious or arrogant, because
Right.
I know that the person I'm really criticizing as much as anyone else is myself.
Right.
Yeah.
Yeah, I aspire to that kind of humility. So let me ask. I actually have been going back through some of your recent blog posts, and I thought a
Oh
good
yeah.
one to ask you about to get things started is your recent lecture on "Is Science Broken?" So that was the title, and this was, I
Oh yeah.
think, yeah, this was South America where you gave this talk.
Um, so the Is Science Broken talk that was in London.
Ah, okay.
I was in Brazil last year giving a rather similar talk, but
Hmm.
this one was the post in question was in London.
I see.
It
Okay.
was based on a lecture in London. Yeah.
Hmm. And so, can you just give us a sense of the gist of your talk? And you could just start by saying, "What's the answer to your question? Is science broken?"
Um, well, actually, that's the hardest part, I think, to answer that particular question. The three-word question: Is science broken? Uh, and in my post, I didn't actually answer that question. I think it's a very difficult question to answer because it could mean anything. I mean, if you say, "Is science broken?" you might mean, is the whole of science so irredeemably flawed that nothing good can come of it?
Hmm.
Which is obviously not true. Um, but that's not the only meaning that's not the only way you could interpret it. So in the post, what I in the post called "Is Science Broken?" What I argue is that the way in which science is done, the way in which, well, large parts of science today are done. So most of neuroscience, most of psychology, some of biology, economics, the way in which these fields approach science is fundamentally somewhat at odds with the ideal of how science should be done,
Hmm.
in the sense that the way in which we feel science should be done is that you have a hypothesis, which is a theory about the world. You make sure that hypothesis is testable, so that there's some way, in theory, that it could be proven false, if in fact it is false, so that it makes predictions that are concrete enough that we can then do an experiment to find out whether it's false. And then we go and test it, and if it is false, then we know it's false, because the data will show that. And if it's true, um, although we can never finally prove it to be true, we can at least show that it's survived many, many tests that have tried to prove it false.
Hmm.
Um, that, I think, is the ideal. That's the way in which many scientists say science should work, but it's not how, in many areas of science, that's the exception rather than the rule. So it
Yeah.
does happen, certainly,
Hmm.
but it's easy to go about science in a very different way. And in fact, scientists are rewarded in many ways for doing this, such as rather than having the hypothesis ahead of time, you form your hypothesis to explain the results you happen to have found,
Right.
Which ensures that your hypotheses can never be tested, really, because they're always one step ahead, if you like, of the data. And then if the data come out and they show that your hypothesis is false, it's possible to continue with your analyses or continue running experiments until you happen to find results that are consistent with the hypothesis, and then publish those and not publish the contrary findings. So I would say that.
Sorry, go ahead.
Sorry.
Well, I was going to ask, it sounds like you're getting into the territory that the statistician John Ioannidis has been
Yes. Yeah. Absolutely.
publishing
Yeah.
on, the unreliability of the scientific literature. I just wonder if when he started, I think his first paper was 2005, and he's published many papers since then. Are the sorts of things that he has been saying about how a majority of peer-reviewed claims turn out to be erroneous? Are
Yeah.
those
Yeah.
surprising to you, at least in the extent of the problem, or is that what you would have expected because of your own experience as a scientist?
So no, I mean, I would say they're not surprising. They're not. They're certainly believable. I mean, so, and John Ioannidis, his claims, I think, are actually almost. I think it's very difficult to disagree with them, because what he does is he simply proceeds from the assumption that positive results are more likely to be published than negative ones,
Mm-hmm.
and that scientists like to publish positive results more than negative ones. And he then shows that even if we assume that most of the experiments we're doing are fairly well designed, but they simply have a limited power to detect even true effects, that if you work under those assumptions, the the almost inevitable conclusion is that most of the positive results that are claimed will not actually be true positives.
Mm-hmm.
Yeah.
Have you? Well, I know you have, but I'll ask you anyway. Have you suggested reforms in the way that science is carried out and published that can reduce this problem?
Yes. So this is actually one of my longest running topics on my blog. I think my very first post was on this issue. So essentially the reforms that I've proposed, well, and many other people have proposed, include pre-registering of research protocols. So essentially, instead of research being done in a kind of black box, as it were, up until the moment you want to publish it, we should publicly announce or pre-register what we're planning to do and what analyses we're planning to run and what results we're planning to be interested in, before then going out and conducting an experiment. This would, although it would be quite difficult, or rather, it would be quite a lot of work to implement this. But it would, at a stroke, pretty much prevent selective publication, selective analyses, producing selective positive results.
That's
and
sorry, keep going.
Oh, sorry. And so, although this is a very major change to how research will be conducted, or rather how research will be published, and when I started out writing about this, so this has already been implemented in the case of clinical trials, but I am of the opinion that this should be rolled out much more widely, including many kinds of research which aren't clinical trials, but which are using statistical tests to examine the significance of effects, which is a very broad remit, and that encompasses large amounts of science. I think that pre-registration would allow all of those fields to be improved and prevent most of these problems.
Yeah, I see. So this would prevent, you know, the term the Texas sharpshooters fallacy?
Yeah, exactly. Yeah,
Yeah, where you say, you know, you get all these results almost at random if you've got large enough data sets that you're looking at, and then you just circle
yes,
the correlations post hoc, as you said before,
yeah,
um, that apparently demonstrate some hypothesis. And so you're
yeah,
saying if you announce your that the idealized way and correct way to do science is to announce your hypothesis in advance, so you can't do that post hoc kind of hypothesizing.
yeah. Well, or rather, so the way in which I would say we should be doing it is that you can do that. You can make post hoc um, hypotheses, but it should just be transparent that they are post hoc.
I see.
So there's nothing wrong with proposing hypotheses after the fact, so long as you're not trying to claim that they were the hypothesis all along,
Sure.
or consciously or unconsciously giving that impression. Um, and so with pre-registration, what happens is that you pre-register your hypothesis, your protocol, and your plans, and then you go and do that, and then you can do anything else you like, and you can come up with any other hypotheses you like. Um, but it's just transparent that those are post hoc, and then there's no sense that they're being confused with or replacing the a priori hypotheses.
Can I ask? I wanted to make sure, and especially now that I know that that if you have a focus, it would be on neuroimaging. I just wonder if you could talk a little bit about some of the problems in neuroimaging, and also if you have anything to say about optogenetics, I'd really love to hear it. I've written a little
Mm,
bit about optogenetics because I read this flurry of stories. I don't know, a couple of years ago, I guess it was, about
mm,
how powerful it was as a way of understanding what's going on in the brain and possibly manipulating the brain to treat mental
mm,
illnesses and so forth. Um,
mm.
so, optogenetics or anything else, MRI, what are some of the common pitfalls of some of these techniques?
Well, I mean that's a big question.
Right.
So I'll start with optogenetics. I actually don't know that much about that, but I think like any other method, it can be overhyped. So as I understand it, optogenetics is actually a very interesting and very useful tool for neuroscientists to manipulate the activity in particular cells, or to measure the activity in particular cells. And that can be very useful for studying, the brains of mice and rats. Um, but I would say there's no way that that's going to directly translate into treating mental illness. Um, I mean, maybe, you know, tomorrow someone will come up with, a proof that I'm wrong about that. But I think it's very unlikely. I think we just don't know enough about mental illness. We don't know enough about how it works in the brain, or how or even how mice and rats, whether they even experience, depression or psychosis in anything like the same way humans do.
Mhm.
So I would say that optogenetics is a very useful tool for basic research, but um, anyone who's claiming that that's going to be like a magic wand curing mental illness, it's just overselling it, and it's raising false hopes, I would say.
And what about MRI?
Mm,
I remember, I started writing about mental illness in the late 1980s for Scientific American, and I remember um, encountering papers. Scientists were really excited about the possibility of finding neurological differences between, let's say, schizophrenics and healthy people, and there were
mm,
all these papers that claimed to have found such
mm,
differences, and of course, over time, none of those claims have really held up.
mm.
I just wonder if you see much potential, if you've seen any robust findings along these lines, neuroimaging actually revealing something significant in the brain about certain kinds of mental illness.
Um, well, I mean, it's difficult to know about that because many of the claims or publications that have come out, only come out in the past few years, so we don't know whether they're going to be proved to be robust or not.
Mm.
So I can't say everything that's been published yesterday. We don't know what's going to happen with that. But certainly, neuroimaging of psychiatric disorders, I would say, has not really lived up to the early promise, in the sense that we're still today researching and searching for robust, replicable differences, um, using MRI, fMRI, which and there are various meta-analyses coming out recently. There was one from, I think, from John Ioannidis, saying that most of the most of the claims about differences in the structure of the brain, uh, which have been made with MRI, and differences in the structure of the brain between people with schizophrenia and controls, for instance, um, pretty much all the differences are explained by publication bias to the false positives. I'm just trying to Google this at the moment now,
Mm.
because I remember I blogged about it at the time. But so I'm yeah, basic analysis of studies of differences in the brain structure, different size and shape of different brain regions in people with various different mental disorders. And this was like a meta-analysis. So it was a meta-analysis of meta-analyses, each of which contain dozens of different papers. Um, so this was 41 different meta-analyses, but they found that there was uh, an increase. There were more
Mm.
positive results than would be expected by
Wow.
if all of these results were real, um, suggesting publication bias. Uh, Actually, I think there was another paper of his on the brain as well. Yeah, so essentially, um, I think there's a lot of problems with publication bias and selective publication in the field, people look for differences between patient and control groups, and then don't publish the negative results. There was another large study recently on autism, which found that there have been lots of different claims about differences in the brain of people with autism, but most of these come from small studies, very small sample size. And there was a paper came out in 2014 using an enormous sample called the ABIDE database, ABIDE database. And this study had 539 people with autism and 573 controls, so which makes it like by far larger than any other study. And they pretty much found nothing, no differences in the size or shape, in the size of any brain region. Even though many other studies with much smaller sample sizes have found differences.
Yeah,
Yeah.
I don't think I'd heard of that study.
Yeah.
Let me just, I wanted to throw an hypothesis out at you.
Yeah.
That I've been toying with a little bit, and maybe it's a truism. It's just crushingly obvious to everybody else, and I just I think it's significant. The idea is that well, it seems to me it would have been a reasonable assumption to think that as we get higher and higher resolution instruments for studying the brain, for studying the genome, various aspects of physiology, that we should understand our physiology better, understand our immune systems better, the connections between genetics and mental illness, between neural processes and mental illness or neural structure and so forth. But in fact, what's happened, and of course, we're getting better and better instruments and also more and more powerful computers to process the data and so forth and look for patterns. And what seems to be happening is not that our minds and brains and various aspects of our bodies are coming into sharper and sharper focus, but that we are getting overwhelmed by data, and it's harder and harder in some cases to find the signal in the noise, and we're much more susceptible now to false positives, to what are actually random correlations to which we attribute significance.
Yeah. So if you take the very same data from 10 years ago in an fMRI study and you look at it now, the number of possible ways of analyzing the data has increased exponentially year on year. And that, I think, has been a very good thing in some ways, but it has increased the number of ways you can make false positives as well. And in general, I think we have made enormous methodological advances, but many of these advances we've kind of rushed into using every new method that comes along. And maybe this is inevitable, but we start to use every new method for doing neuroscience as soon as it becomes available. And then we discover 5 years later that we've made, a fundamental mistake or in interpreting it, or some kind of technical problem. And then 10 years later, we've solved that problem, but perhaps another problem has arisen. So I think, and I've heard people say that, I think someone told me once, the first 10 years of use of any method is just like testing it out, and you shouldn't take the claims too seriously. It's just ultimately just a way of validating the method. So I think that we've got many new methods. We don't really understand yet how useful they are or what the limitations are. But with every new method, and particularly with every new method of analysis, there's an increased risk of false positives as well.
Yeah. Okay, I wanted to stick with psychiatry a little bit,
Oh yeah,
treatments for and go to treatments for mental illness.
oh
So
yeah,
let's say, I don't know, psychopharmacology,
yeah,
and we've gone through this whole wave of enthusiasm for SSRIs and other drugs for treating mental disorders.
yeah.
SSRIs were marketed along with theories that supposedly showed that depression, for example, stemmed from some kind of misregulation of serotonin in the brain and so forth. And
Mm.
I just wonder if you can talk about that whole application of neuroscience.
So this is um an interesting question, and I think again it was a question of prematurity. Um, we are still every day learning fundamental new discoveries about, let's say, serotonin or any other um, neurotransmitter. Um, so we really don't understand fundamentally what serotonin, or in fact, pretty much any transmitter does in the brain, what its role is. Um, and we're learning every day about how serotonin um, serotonin neurons, let's say, operate. Um, so it was just discovered recently. This is sort of a good example that I like um, to quote. It was discovered recently that serotonin neurons, in many cases, also release another transmitter, glutamate. Um,
Mm-hmm.
so uh, this and this actually fundamentally goes against the whole idea that every neuron releases a single transmitter. Um,
Hmm.
and I'm just looking for the paper here. Yeah, and actually there's been a number of studies recently showing this. So many serotonin neurons aren't actually only serotonin neurons, and for many years that was, believed to be absolutely a sort of fundamental truth that every neuron has a single transmitter and it only releases one thing. Um, but it's recently been shown that many serotonin neurons release glutamate, and glutamate is a completely different transmitter, and it in itself has is very poorly understood. Um, and I think overall, what this makes me just, I think should, and I think um, what it should emphasize is that we really don't understand how any given transmitter works, and there's just so much that's unknown. So to say that, yes, to say that depression is caused by a deficiency in serotonin because serotonin causes like happiness, it's the happy molecule. Um, that's just going far, far, far beyond what we know at the moment. But of course, if you're going to sell a drug, then you want to have a simple narrative. But I think we just don't know yet. So I think it's too early to say. In my personal opinion, it's too early to say whether serotonin is or isn't associated with depression. Um, in some way, we just don't know enough.
Um, I can imagine what you think about some of the popular coverage of oxytocin, the love molecule,
Yeah, yeah,
or mirror neurons, or I mean, there's so many things I could ask you about.
yeah,
Um,
exactly. Oxytocin. I often have said it's the new serotonin because it's pretty much the same scenario again, but just like 10 years later, 20 years later, in that, serotonin used to be said it was a very simple serotonin equals happiness. And it's become clear that it's much more complicated than that. But now people are saying oxytocin equals, well, I don't know, trust or love or hugs or something. But I don't. I think also with oxytocin there are a lot of technical issues that remain to be resolved. So many of the studies on oxytocin, they use a nasal spray of the oxytocin molecule, but it's really an open question as to whether that oxytocin actually reaches in the brain at all. So there are very few studies. In fact, there's only one study to my knowledge that has ever looked at the fundamental question of whether nasal oxytocin can actually reach the brain in humans. It's been shown that it does in mice, but it's different in humans.
Huh.
And the only one study that looked at this found that it did, but only 75 minutes later after the spray,
Huh.
not after 45 or 60 minutes, which would kind of be a very serious problem, because many studies have used oxytocin less than 75 minutes for the tests that they've done. But also the sample size was absolutely tiny. It was like 4 people in each group, and one group had there was a group which only had one person in it. It was quite a complicated design, but the study was absolutely tiny. And I blogged about this. This was in 2013. The question: does nasal oxytocin into the brain? But the point is that I think it's an open question whether it does, but we still don't even know that fundamental, really, really fundamental question. And so it's really presumptuous to say, pretty much, to make grand claims about anything else.
We've actually we don't have that much time left, so I have a few big questions that I wanted to be sure to
Oh,
ask
sure.
you.
Yeah.
So one would be the whole quest for a theory of consciousness. You know,
Yeah.
the mind body problem goes back thousands of years.
Yeah.
Plato and Aristotle contemplated it. How
Yeah.
matter can produce this subjective phenomenon of
Yeah.
subjective awareness and so forth. And
Yeah.
this is the sort of thing that Christoph Koch has pursued, and many, many others. Francis Crick was fascinated with the possibility of a theory of consciousness. I just
Yeah.
wonder where you think, if you can give us any sense of where we are in that quest, and whether even it will be uh, fulfilled ever, or whether it might always be open-ended in some sense.
So so I'm not this is not a specialist topic of mine, but certainly I've never seen anything which suggests that we are close to solving that. And I don't know where a solution would come from. I mean, I suspect if we ever did solve the hard problem of consciousness, the solution would be from something like computational theories, computational modeling, and computational computer science, really, explaining how many trillions of synapses can, that kind of some theory of how something can emerge from trillions of different interacting units is my impression, but fundamentally I don't know.
Have you encountered one thing that distresses me, and to me suggests that the field, far from progressing, is maybe taking a step backwards, is that Koch himself has recently in Scientific American espoused an idea called panpsychism, which suggests that
Oh yeah.
consciousness is a
Yeah.
uh, attribute of possibly all forms of matter. That rocks have some kind of rudimentary uh, consciousness, or anything that could be said in some way to process information, whatever the hell that is. And that seems to me to be just like, I don't know, mystification, and a sign of desperation.
So yeah, so certainly I don't think that gets us any closer to answering what you can say maybe rocks have a consciousness, but then you're stretching the definition of the word consciousness
Right.
probably to the point that it's not really helping us to understand, because what we're really interested in is what makes human consciousness happen, what makes our own the complexity of human consciousness possible, and
Yeah,
at the moment we don't know.
one way that I thought um, that some of these really deep philosophical issues could be solved, and lots of practical issues as well, is if neuroscientists can discover the neural code, whatever set of rules or algorithms
Mm,
might
mm,
transform neural processes, the ebb and flow of neurotransmitters and action potentials, into the thoughts that we're having right now as we have this conversation. Have you
yeah.
seen is the neural code problem something that you're interested in? Have you seen any particularly interesting developments in neural code research?
So that's not one of the topics that I follow actively. It, but I think yes, it's still something we don't really know the answer to. I feel that solving, yes, as you say, solving that, cracking the neural code would probably be the only way to make progress in the question of consciousness. I'm not sure how to go about that. Well, I think that this was what the Human Brain Project was trying to do, but it has turned into a bit of a fiasco.
Oh, good. I'm glad you brought that up.
Yes.
That was one of my big questions. Yeah. What about both the European Human Brain Project and the American Brain Initiative? There's been a lot of
Yeah.
controversy about whether they're premature or too top down. What do you think about them?
So, from as I understand it, there's a there's a big sort of transatlantic difference, and that the Brain Project is obviously in the very early stages, but it seems to be better organized and better led. A lot of people, a lot of European neuroscientists who were involved in the Human Brain Project have kind of revolted, uh, and hundreds of people signed the petition last year, the Neuro Future petition, and are very, very unhappy about the organisation, the priorities that they're setting, and the fundamental sort of scientific problem that has emerged is that they're trying. The whole idea is that you can simulate the brain using computational models, um, from the bottom up. So you can simulate a single neuron, and then if you scale that simulation up and simulate like a million neurons, then maybe you'll simulate the brain. But there's a, I mean, there's a very strong argument that we just don't know enough about how the brain works, and even about the very simplest level, we just don't know enough about the basic neurons to do that.
Yeah, I mean, because
So this bottom up approach,
you have to make assumptions at every level of what
yeah, exactly,
to
yeah,
include and what to leave out. This gets back to the neural coding
yeah,
problem,
yeah,
where, I mean, you know, the last time I looked at it, there were all these different versions of the neural code that said that the important stuff, the important information processing was happening at all these different levels, both from within individual neurons way up to this meta scale of massive populations of neurons interacting with each other. And there's no way of
Yeah.
knowing at this point which of those levels is the right level, or maybe they're all right. Maybe they all have to be included.
Mm. But yeah, exactly. So, but you can't get anything right if you've got a bottom-up model and your fundamental, the bottom level is not accurate, then the whole thing won't work. Because if even the slightest difference, even the slightest inaccuracy in your modeling of the lowest level will then sort of snowball up the levels.
So, I mean, if you were the king of neuroscience, would you disband the Human Brain Project?
Well, a lot of people have pretty much proposed that. A lot of people who have been involved in the HBP have said pretty much that unless there's some, transformative, game-changing improvements in the way it's run, that the money ought to be broken. The money ought to be distributed amongst individual neuroscientists, in the form of grants for individual projects, and that it's too early to do this kind of grand monolithic approach.
Right.
But then,
I'm sorry, but did you also say that the Europeans think that the Americans are doing a better job of creating this kind of big project?
that's the impression I got, and in fact, I did so I interviewed Zach Minen,
Mm-hmm.
or last year, and he was the sort of main ringleader of the protests against the HPP. He was involved in the HPP, and I asked him, what do you think about the Brain Initiative? And he said, I think the US made almost all the right moves. They chose a committee. They got advice from everyone involved in the Human Genome Project. They had a broad range of respected neuroscientists, an open and flexible process, and a competitive grant structure rather than a sort of dictatorial decision-making structure. And he says Europe could learn a lot from their example. So yes, the impression I get is that the Brain Initiative has not made the same problems on a sort of organizational level, but it remains to be seen about the science.
All right. Well, let me tell you one really big concern that I have with the Brain Initiative here in the States. It's that
Yeah.
more than half of the money comes from the Pentagon, and and that is it's not a secret, but it's something that has been discussed little or not at all by journalists or by neuroscientists themselves. And it seems to me that it's quite significant.
I didn't know that, actually.
Yeah, it's the Defense Advanced Research Projects Agency that's providing more than half of the money. Also, and in part, I think because of the Pentagon emphasis, there is particular interest with applications, including brain implants. Electrodes that are inserted into the brain to stimulate various parts. These are the kinds of things that can be used to restore function to people who've been paralyzed or had brain trauma and this sort of thing. One of the people whose work was mentioned in a story on these applications is a neurologist named Helen Mayberg, who has worked on
Yes.
implants for depression.
Depression. Yeah,
Yeah, and I, and whose work has gotten, I think, way much more positive attention than it deserves. So,
yeah.
yeah, so those are some of the things that I worry about our particular supposedly superior initiative.
Mm.
So,
Well, I mean, I suppose it depends on how the money is spent.
yeah,
If the projects that they're going to be spending, that they're going to be funding, are
yep,
really going to help us understand the brain, or are they going to be militarized
uh,
application?
right. Um, so there's actually we're close to running out of time.
Okay,
I had one other question I wanted to ask you about, kind of
sure.
a blue sky science fiction-ish question. Are you familiar with the
Okay,
term
I like those.
the singularity?
Yes, yeah.
Okay, so I just wanted to give you a chance to, speculate all you like, and talk about this possibility of humans becoming super intelligent because, let's say, we know so much about the brain that we can soup our brains up with brain implants, become bionic, and maybe even turn our psyches into giant software programs and download them into computers and live forever, that kind of thing.
Mm.
So,
So
what do you think about the singularity?
personally, I think that the one day it will be possible to create an artificial intelligence which is, well, you'll never know if it's sentient, but which seems to be sentient, um, and that will be a transformative moment. But I don't think we're really going to be able to upgrade our own intelligence. So the singularity might be that we created an AI which is able to do things which we can't. But then the problem might be that we'll be left behind. I think the brain, I think the idea of uploading consciousness from the brain is just, I don't think it's going to be feasible. You could create perhaps consciousness from scratch in an artificial sense. I think there's nothing that would stop you from doing that. I mean, after all, it's not that hard to create consciousness because, millions of conscious human beings are born every day and they start from an egg and a sperm. So, it can't be that hard to actually create consciousness. To upload consciousness is like, I'm not even sure how you do it. You'd have to investigate. You'd have to measure you'd have to measure the brain in such amazing detail. There's no way we can do that and then digitize that information. But we couldn't scan the brain in that much detail. I don't think it's going to be possible.
Yeah. Okay. Then, actually, now I have one more question, just following up on
Okay.
what you just said. Um, what about the possibility of a technology that can really read minds? That
Hmm.
you can put somebody in some kind of scanning device
Hmm.
and not only know whether they are happy or sad or schizophrenic or
Yeah.
those sorts of things, but actually,
Hmm.
let's say if it's a terrorist or a suspected terrorist, you can peer into their mind and find detailed plots for blowing up people and buildings and so forth. How feasible is that sort of technology?
I think it is feasible in theory, but it would require us to understand the neural code much, much better than we do. So there have been various people who have sort of like claimed we're almost at that stage already, and I don't think we are anywhere near that stage. But I think it will be possible in theory. Um,
Are you going to live long enough to see it?
may I? Yeah,
Do you want
I'd
to live
lean
long enough to
towards.
see it?
Well, yeah. Well, as a blogger, I do because then I could blog about it, and I could say brain brain imaging, brain reading is here. But what does it mean?
Right.
But I'd lean towards saying yes, but well, I don't know. Maybe I think it will happen one day, but whether it'll be 50 or 100 or 200 years, I'm not sure.
Yeah. And I hope the technology is used wisely by the leaders of the future if it is invented.
Oh yeah.
um,
Well, yeah. It might. Mm, sorry.
so okay.